Welcome everyone. Let's discuss another question from GATE 2020 Biotech exam. The question says, assume that the cell culture was started with 5 human fibroblast cells. 2 cells did not divide even once, whereas the other 3 cells completed 3 rounds of cell division. At this stage, the total number of kinetochores in all the cells put together. So, first we need to understand what all information is given here. So there are total 5 human fibroblast cells. These are the cells which are responsible for the formation of connective tissues. And total 5 human fibroblast cells are there, out of which 2 cells did not divide, while 3 cells completed 3 rounds of cell division. We all know after each round of a cell division in mitosis, we will get 2 identical daughter cells. So at the end of 3 cell division, the total number of cells are going to be 2 raised to the power n. N is the total number of division, which in this case is going to be 3. So total 8 cells. If I'm starting with 1 cell, but remember we are starting with how many cells? 3. So 24 cells. Also, we need to consider 2 cells which did not divide even once. So the total number of cells are going to be 24 cells plus 2 cells, which is going to be 26 cells. Now, what we need to find out is the total number of kinetochores in all the cells put together. So we need to figure out the total number of kinetochores which are present in these 26 cells. So first we need to understand what is kinetochore. So these are the protein complex which is formed at the centromere. So this is the point where the kinetochores are formed. And what is its function? It attaches the duplicated chromosome. So what I've shown here is a chromosome. It's a single chromosome only, but consisting of 2 sister chromatids. During the mitosis, what will happen? These 2 sister chromatids will move towards the opposite poles, and at the end of the mitosis, we'll get 2 identical daughter cells carrying each chromosome. Now, what we need to understand is the different phases of the cell cycle. So we have the G1 phase, S phase. And why this is important? Because we also need to understand how many kinetochores are going to be there at different phases. So we have G1, S, G2, and then we have M phase. So at G1 phase, what we have is a chromosome which has not undergone DNA replication. Now, during the S phase, the DNA replication takes place, and this structure gets converted into a chromosome with a 2 sister chromatids. We need to focus the total number of chromosomes are going to be the same throughout, which is going to be 46. But when we compare the G1 with the S phase, this chromosome is carrying the 2 sister chromatids. So the amount of DNA is double. As we all know, the DNA replication takes place in the S phase. Even during the G2 phase, this structure is going to be maintained. During the M phase, we have prophase, metaphase, anaphase, and telophase. So at the end of M phase, at the end of telophase, we'll get 2 identical daughter cells carrying equal number of chromosomes. Now, look at the structure of this chromosome. These 2 sister chromatids will again get separated and we'll have 2 identical daughter cells. So I'm calling this as a chromosome, even this as a chromosome, the only difference being the amount of DNA is doubled here in the S phase. And at the end of the M phase, this will result in the generation of a 2 identical daughter cells having a single chromatid. So let's have a look at this diagram. This is what I was talking about during the transition from the metaphase to the anaphase. This sister chromatid, so this is a single chromosome. Total, there are 3 chromosomes, and you have the centromere here at this place, and the red ones are the kinetochore complex, which is attached to both the 2 sister chromatids. It helps in the formation of a kinetochore microtubule shown in green. And these sister chromatids will pull towards the opposite poles, which you can see here. And once the cytokinesis takes place, you'll have 2 identical daughter cells carrying the equal number of chromosomes. Now, one thing we need to keep into our mind that at this place, this chromosome is carrying two sister chromatids and hence the two kinetochores. In this particular case, and once the cell has completed division, you'll have a single kinetochore for a single chromatid. So let's solve this question. We have calculated total number of cells, 26 cells, and a single cell is going to carry 46 chromosome, and hence the 46 kinetochore, because there are going to be 46 chromatid. Why I'm considering the 46 chromatid? Because remember, it's given the three cells completed 3 rounds of cell division. So they have completed the M phase and have moved to the G1 phase. So this structure exists during the G1 phase, and you have a single kinetochore for a single chromatid. So 26 cells into 46. So the final answer is going to be 1196. I hope this answer is clear to each and every one of you. If you have any doubt, please feel free to comment. Thanks for watching the video.